fantastic introduction. Welcome back to Colloquium. We have two much more interesting talks to hear than me explaining what's going wrong with the database today, thank goodness. So we'll be hearing from Auguste and from Megan, and Audrey's here, I think, to do our first introduction.
Yes. All right, so it's great to be here in person. This is my first in-person student summer colloquium of the year. It's so refreshing. So I am very pleased to introduce Auguste Dutcher, and Auguste has enough results for a full hour talk, so I'm going to be super short. Auguste is a senior student in the genetics program. She is fantastic. She is an integral part of our close-knit lab, and it is a pleasure working with her. I feel very, very lucky. And so some of you may remember Auguste's talk from last year, which is a very cool project and is not what she's talking about today. She has this completely independent project that is also very fun and exciting. And with that, I'm just going to turn it over to Auguste.
Great. Can you all hear me okay? Okay, I can also fire up the mic if need be, but thank you very much, Audrey, for that introduction. And just to reiterate what many have said, also very delighted to be here in person. This is my first ever in-person colloquium session. So the work I'm going to be presenting today is different from last year, but a continuation of some major themes of interrogating the RNA binding protein Ssd1's role in aneuploidy tolerance. And so, want
to let Ellen in actually?
Oh, Ellen's what?
Do you have that? Admit.
Oh, okay. Oh no. Okay. Hi, Ellen. You're in now. Great. Okay. Sorry, I have to now bring my cursor back over here. Okay. So. Wonderful. So as you might have guessed, very central to my work today that I'm going to be presenting today are questions around aneuploidy, which, as you're all familiar, is broadly defined as an imbalanced set of chromosomes, but which I'm going to be discussing today specifically in the context of chromosome amplification. Like I'm showing in this cartoon here, specifically today, I'll be talking about haploid genomes with a single chromosome amplified, and specifically tolerance of that amplification. So many of you might be familiar with aneuploidy and in particular, its relevance for human disease. So, for example, most aneuploidies are inviable during human development, and that's why you've probably only heard of a handful. On the other hand, aneuploidy is incredibly common in cancer. So by some estimates, over 90% of solid tumors are aneuploid. So I'll note here that in this case, this is providing a proliferative advantage often from the perspective of the cell. So despite the importance of aneuploidy and its relevance here for pathology, and I think basically as a very interesting set of biological questions all on their own, experts in the field are really still interrogating the cellular consequences of aneuploidy. So what I mean by that is what kinds of stresses does aneuploidy really represent, and how do cells handle that, which really feeds back into my question about aneuploidy tolerance. So my entree into this project, as I'll explain a little bit more in a moment, kind of has a little bit of a mystery as its centerpiece, and that is what a particular RNA binding protein, Ssd1, is doing to facilitate aneuploidy tolerance in wild yeast. So in many ways, I'm looking at this super tiny piece of a much broader puzzle. And so one way to frame this work that I think is pretty interesting is to ask, what can exploring the role of this one RNA binding protein tell us about aneuploidy and aneuploidy tolerance more generally? And what can interrogating the cellular stresses that are imposed by aneuploidy, what kind of clues can that give us about why Ssd1 might be important? So in other words, rather than thinking about these as two discrete sets of questions about the consequences of aneuploidy, and then specifically what this protein is doing, I really like to think about this as a conversation about how can these two sets of questions inform one another. So that's something I encourage you to keep in mind. So as some of you may be familiar with, the foundation for this work really came out of some fascinating observations out of Audrey's lab a number of years ago at this point, noticing some really interesting differences in the ability of yeast to tolerate having extra chromosomes. So for example, this wild strain of yeast, YPS1009, which is isolated from oak soil, is naturally aneuploid. That is to say, it's aneuploid when you find it in nature, and it appears to tolerate this aneuploidy relatively well. Whereas there's a commonly used lab strain called W303, where single chromosome amplifications were reported to be extremely toxic and result in pretty severe phenotypes, including slow growth, respiratory defects, and evidence of protein aggregates. And so, long story, lots of work short, Audrey and her team mapped the genetic basis of this difference in aneuploidy tolerance to a single protein called SSD1, this RNA binding protein, which, as it turns out, is present in its full length, fully functional form in this wild strain YPS1009. But W303 contains a truncated allele that's only partially functional. And so indeed, what her group found is that loss of this RNA binding protein sensitizes cells to aneuploidy. So I'm just showing one quick example of that here, where what I want to point out is that in euploids, in other words, where there is no chromosome amplification, you can delete SSD1 and cells grow comparably to their wild type counterparts. Whereas when you duplicate chromosome 12 and you delete SSD1, you see this dramatic reduction in growth rate relative to the wild type aneuploids, indicating this SSD1 dependent aneuploidy sensitivity. So there's been a ton of work that's happened in the lab since then, and I hope you'll all have the opportunity to hear about that at some point, about the myriad of projects that have grown out of this foundation. One important piece of that I wanted to mention today is that we have since undertaken pursuing a really exciting set of genetic tools. And that is that James Hose in our lab last year started to go beyond looking at chromosome 12 and a handful of other aneuploids that we had examined and characterized in the past, to go ahead and undertake this incredible task of constructing every viable chromosome duplication across budding yeast's 16 chromosomes. And so he, along with a fantastic new postdoc in our lab, Julie, have worked to characterize the growth of these strains and have constructed every viable aneuploid along with its corresponding SSD1 deletion. And so what Julie has done that's super interesting is to plot the relative growth rate of each of these new aneuploids against, in this case, the number of open reading frames contained on the corresponding duplicated chromosome. So on one axis, we have increasing gene content, relative growth rate on the other. This is part of a much bigger body of work. But the point I want to make here is that when she plotted the growth rate of the wild type aneuploids, she was able to explain a pretty decent portion of the variation of growth rate, just specifically on based on this number of open reading frames alone. But the piece of this that's most interesting and most relevant to me is that when she plotted the same data, but for the aneuploids that have SSD1 deleted, we again see a pretty good explanation of the variation in growth rate by ORF number, but also notably a steeper slope of this line. And what this suggests to us is that the cost of chromosome duplication is higher when SSD1 is absent. Of course, a super interesting observation, but it doesn't really tell us why this is the case. And this why has been sort of the basis of my projects since I've started grad school. And so SSD1 has received, as we usually say, a modest amount of study in the past and has been implicated in really a pretty wide range of functions. Potentially most notably cell wall integrity via binding to cell wall transcripts that are localized to the bud neck during active growth. SSD1 has also been implicated in translational silencing of some of its target transcripts and in localization of mRNAs to P bodies under stress conditions. And then in other screens has been associated with cell cycle progression and quiescence. So we know that it's implicated in many processes, but again, we don't know which of these functions are important for aneuploidy tolerance. So last year I spoke to you a bit about genetic interactions in SSD1's regulatory network. And in this case, I was probing single gene knockouts. So to take kind of a different approach, rather than the specific genetic approach, a broader genomic approach to the similar sets of questions, we wanted to find out what we could learn from a comprehensive or nearly comprehensive gene overexpression experiment. And that's what I'll be talking about today. So the central questions for this project were very broadly, what can we learn from gene overexpression about SSD1 dependent aneuploidy toxicity? Specifically, some insights I was hoping to get from my data were which genes uniquely affect the growth of aneuploids that lack SSD1, and on a related note, which genes are particularly toxic when overexpressed in an SSD1 mutant background. And again, these insights, I think, are particularly informative to that first question of what can we learn from these toxic genes about models of SSD1 dependent aneuploid sensitivity. So the tool that I used to do this is called the MOBI 1.0 library, which was developed out of Charlie Boone's lab at the University of Toronto. And what this is a plasmid library that consists of centromere-based, so low copy number plasmids, each of which contain an open reading frame plus the native promoter and terminator of each of about 4,500 genes. So for those of you not familiar, that makes up a very decent proportion of the genes expressed in yeast, in budding yeast, I should say. And importantly, each of these plasmids harbors a unique oligo barcode, which is super useful for me because when I am doing sequencing to assess plasmid abundance or overexpression of particular genes, I need not sequence the entire gene, right? I can just sequence the barcode and know what's there. As far as my experimental design, I leveraged this suite of strains where I have every combination of with and without SSD1 and with and without aneuploidy. In this case, an extra copy of chromosome 12, which is our best characterized strain to date. I use selection to maintain the aneuploidy across the course of the experiment. And so the way that this works is essentially after transforming the plasmid library into these strains, I sequence the starting pool. So I understood the pool of constructs that I was starting with, grew the pool for 10 generations, and then sequenced them again at 10 generations. So in this experiment, the idea is that we have, again, a single gene overexpressed in each individual cell. And so the principle is that barcodes that correspond to genes that are beneficial for cells should increase in abundance over the course of this 10 generations, whereas barcodes that correspond to genes that are detrimental should decrease in abundance over time. So in my cartoon example here, the yellow cells harbor a plasmid which has a gene which, when that cell has an extra copy, it does better. It has an advantage over its competitors. And so it increases in abundance in the pool over time. Whereas this blue cell is harboring a construct with a gene that is detrimental for growth of that cell, at least relatively speaking, it decreases in abundance over time. A quick note about rigor and reproducibility in this type of experiment. So I mentioned that we used, first of all, drug markers to maintain the aneuploidy over the course of the experiment. So these were only strictly necessary for the aneuploids because each copy of the duplicated chromosome contains a different drug marker. But of course, we also engineered our euploids to contain identical markers so that we could grow all of the strains in identical conditions across the experiment and making sure we're really only varying what we want to, which in this case is the presence or absence of SSD1 or the presence or absence of aneuploidy. And the second thing I'll mention is that you could imagine that When I transform this plasmid library in my starting pool, just by chance, I might end up with much greater representation of some plasmids over others. And that could be in a systematic way, but or it also could be in a more or less random way that could affect the results of the experiment. So I also conducted multiple independent transformations of the plasmid library to at least start at trying to getting at controlling this potential stochastic starting abundance. And so to jump directly to my results, this is what they look like. This is a full heat map showing all of my results where every row in the heat map effectively represents a different gene. And every tile represents a log fold change of barcode abundance after 10 generations versus that same barcode's abundance at time 0 or the start of the experiment. And I might shorthand this later in my talk to refer to this as a fitness score. And the idea here again is, so if a gene is shown in yellow on the heat map, that means it increased in abundance over time and was beneficial. If it's blue, it decreased in abundance over time and we're inferring that it was detrimental to growth. So in the group separated by the gray bars, I'm showing my 4 different strains. And then there are 4 replicates represented for each strain, 2 from each of 2 independent transformations of the plasmid library. So one thing that I really like about this experimental design is that it allows me to ask specifically what is different about the aneuploid mutant that stands out from either an isogenic euploid, so it's the same, but it doesn't have the chromosome duplication, or an aneuploid wild type, which has the aneuploidy in common, right, but has SSD1 intact. So it really allows me to narrow in on what is different about this strain. And so what might SSD1 be doing specifically in an aneuploid context that's unique from its role in a euploid and is also unique from aneuploids generally speaking. So the first result I wanted to share with you is something that stood out to us right away, which was first that it looked like there was a group of genes that actually selectively benefited the aneuploid mutant relative to any other strain. And many of these genes had shared functions. So I think this is a little bit interesting of a result in and of itself, just that despite already harboring additional gene content in the form of an amplified chromosome, an entire amplified chromosome, there were still genes whose overexpression were able to benefit specifically this strain. And I think this is all the more compelling because they do have a number of shared functions. So these include regulation of translation, mRNA processing, lipid metabolic processes, Golgi vesicle or transport or Golgi apparatus more generally, and regulation of cell cycle or being associated with sites of polarized growth. And so what this suggested to us is that SSD1 deletion might exacerbate aneuploidy stress due to defects in, first of all, translation, maybe of transcripts or proteins that are particularly hard to handle for some reason. In any of those processes related to trafficking or lipids that I mentioned above, and in cell cycle progression, which had been previously noted for aneuploids, or potentially establishment or maintenance of cell polarity during growth and division. So that's one interesting insight that's come out of this work so far. And I think these all represent potential interesting areas of follow-up where we can interrogate if any of these processes are perturbed specifically in aneuploids that lack SSD1. The second really interesting insight that comes out of this experiment is that we have the opportunity to essentially interrogate the effects of aneuploidy at a single gene level. And the reason I think about it this way is because when we introduce a single extra copy of a gene into our euploid cells, we're essentially mimicking what happens when that cell is aneuploid, but just on a gene by gene basis. I think this is super cool and just a cool set of information we can get out of this. I thought it was too cool not to share. So one thing as an example that we can do with these results is actually take those fitness scores for those 2 strains, the euploids that have SSD1 and the euploids that don't, and sum up the fitness scores across across entire individual chromosomes. And then Julie, who is doing that modeling of aneuploidy toxicity I was talking about before, can then incorporate that into her model of growth rates, so that instead of number of genes on the x-axis as we had before, we actually have an aggregate fitness score that's coming directly from the data that I represented here. And what I want to point out about this is that when she plots these growth rates against these aggregate fitness scores. So again, this appears to explain a pretty substantial portion of the variation that we see in relative growth rates. And it does in fact do a little bit of even of a better job in explaining this variation than just the number of genes alone. And the reason that I point that out is that this is really pointing to that the beneficial or perhaps particularly toxic nature of specific genes might be particularly important for understanding SSD1 dependent aneuploidy toxicity. And so this leads me to sort of my next set of results that I wanted to talk about. It really makes me want to know what those genes are that are especially toxic when amplified in an SSD1 deletion background, but are not equally toxic in the same background that has SSD1 intact. And so I'm able to ask what genes essentially are especially toxic when overexpressed in the absence of SSD1. I identified 134 genes that fit that criterion. So I'm only showing genes here that are detrimental. So you see all the fitness scores are negative. 134 genes that are specifically detrimental in euploid SSD1 deletion strain. So I am dying to know what these genes have in common, and I am still working on figuring that out. But in the meantime, we were also super interested in the effect that seeing what the effect would be of restoring any of these genes to euploid levels and what effect that would have on SSD1 dependent aneuploidy sensitivity. So far I've done that with one gene called CCW12. I started with this because, as you can see, it's highly toxic, specifically when SSD1 is absent. It's also importantly a known SSD1 target, and it's encoded on chromosome 12, which again was which corresponds to our best characterized aneuploid to date. And so in the growth rate data on the left, I'm showing what happens when I restore CCW12 essentially to euploid levels in an otherwise aneuploid background. So I'll draw your attention here particularly to the aneuploids. So we have chromosome 12 duplicated. And we can see that in the absence of SSD1, we see this drastic reduction in growth rate that we associate with SSD1 dependent aneuploid sensitivity. However, when I delete one of the now 2 copies of CCW12 that are present in this strain, we see that restoring the copy number of CCW12 to euploid levels largely mitigates this SSD1 dependent sensitivity to chromosome 12 duplication. And so again, I think this result is super interesting because it could represent a really important handle to investigate what is it about this transcript that's making it so toxic, specifically to cells that are lacking SSD1, even though it doesn't affect corresponding euploids in exactly the same way. And so as you might be thinking or noticing, really this, as it always does, leaves us with more unanswered questions than answered ones. And so what we're working to do now is really to sort of reconcile these insights and to create a working model of how SSD1 mediates aneuploidy tolerance. So as a reminder, some of what I've showed you today, we've seen that functional relationships among beneficial genes point to potential mechanisms by which SSD1 mediates aneuploidy tolerance. And I put general with a question mark there because it's an open question about whether those beneficial genes are specific to chromosome 12 or represent more general mechanisms of handling aneuploidy. And I'll be interrogating that in the really near future by repeating this MOBI experiment with some of those other aneuploids I mentioned we now have in hand, which is fantastic. You also saw that from Julie's work, the aneuploid growth is pretty well modeled by the additive effects of single gene overexpression. But then potentially, not totally at odds, but potentially interesting related to that is that I also showed you that SSD1 mutant cells are highly sensitive to amplification of very specific genes. So on the one hand, we're seeing that you need the additive effects across the entire chromosome to model well the growth rate that we're seeing of the aneuploids. And on the other hand, we see that there are specific genes that might be super, super important for causing this SSD1 dependent aneuploidy toxicity that we see. So we're still working on how to reconcile these insights into a model that makes sense. One idea I'll throw out there is that SSD1 might play a key role in managing specific gene products that are super toxic when overexpressed for some reason, but that a defect in management of those very particular genes actually sort of throws off the balance of everything, throws off the threshold of proteostasis tolerance, thereby sensitizing cells to many chromosome duplications. So it'll be interesting to see if I can find other genes that have the same effect, a similar effect as CCW12, and to what extent the effects are additive versus sort of pushing over a line into, okay, now we're super sensitive because we can't handle too much of this sort of one bad thing. And so, as I mentioned, how to reconcile all these insights is still an open question, but I think so far this work has yielded some really potentially useful handles for follow-up to interrogate both how SSD1 is facilitating aneuploidy tolerance, as well as understanding models of SSD1 dependent aneuploidy toxicity more broadly. And with that, I have to give many acknowledgements. Not only would this work not be possible without the collaboration of everyone in my lab, and particularly those highlighted here, but it would be substantially less fun. And so I can't give enough acknowledgement there. Also to my funding sources, particularly the GSTP, who's funded me this past year, Dave Schwartz and Louise Pape as well for their work in coordinating that, and to my committee. Thank you. Chris.
I have two related questions, which is it possible to look at haploid versus diploid differences in SSD1 solutions?
Yes.
Do those show any similar expression?
You mean any similar sensitivity or any like?
Is the differential expression in aneuploid similar to the direction of differential expression in haploid?
I haven't done any comparison of diploid versus haploid expression profile. So, but you mean how they handle having the extra, like how having the extra chromosome affects? Yeah, I'm not sure between the haploids and the, I haven't done that. Yeah.
So many related questions. Do you know much about the conformation of the chromosomes? I know like you could do like a proximity ligation assay, like a IC or CCC. And is it possible that having aneuploidy disrupts that in some way? Like it's not like the rest of the genome isn't accounting for trans effects, but the duplications are interacting physically in some way?
Yeah, it's an interesting idea for sure. It's not something that we've investigated, but it's a super interesting idea. Yeah. Great. Kevin?
Yeah, is SSD1 in the Moby library?
It is not.
Okay.
It is not. No, it would be a great control. Nathaniel?
It might be tricky to disentangle the correlations with growth rate across the different aneuploid chromosomes. Because one thing I'm remembering is that the number of open reading is probably really highly correlated with total chromosome length, in your very gene dense genome. And then your sum of spores, even if every gene had the same average fitness score, you wind up seeing the same correlation you saw before.
Yeah.
So I'm kind of curious whether the fit is truly much better when you have your individual fitness scores or not.
No, that's an absolutely great question. And this is largely Julie's work, who's working with the modeling, but that's exactly what we have been wondering about. One thing that I do think is super interesting is that she found that, for instance, if you exclude the beneficial genes from that aggregate fitness score, in other words, you take out all the positive ones, the fit doesn't get any better. In fact, it gets worse. So I don't know if that really means that we're looking at like, I take your point about, I think that's why it's important to show that the correlation really is quite good, even just with the number of ORFs, because it's possible that the Moby score isn't adding that much, but I still think it could be super interesting.
Well, you can always just randomize the scores across chromosomes to test whether the actual genes that lie on each chromosome are relevant to the fit and not just the number.
Yeah, that's a great idea. Yeah, cool.
Move on to our second talk.
Okay, this is yours, right? Yeah. But then we need to make sure that we see the.
Do you need to plug in? You're at 13%.
I will. Yeah, I'll do that as soon as.
Yeah. Well, maybe I'll get started so we can not get too delayed here. So it's my pleasure to introduce Megan MecKeon. Megan is not in my lab. She's in Christina Hall's lab. She's in the Department of Biomolecular Chemistry. And Christina couldn't be here with us today, but I'm on Megan's committee, and so I'm happy to sub in. Megan has been here since 2018, and before joining UW Genetics, she got her degree in integrative biology from the University of Illinois at Urbana-Champaign, where she studied pineapple genomics in Ray Ming's lab. And she spent actually a couple of years in industry as an analytical chemist, which I'm sure is helpful in her current work. Megan's been studying how Cryptococcus spores work, how they develop and remain dormant and then stop being dormant and germinate and start growing, which are all really critical phases of the life cycle of this organism. And she's been using some really quantitative and high throughput and rigorous tools to do that. And in fact, Christina wanted me to let you know that, in fact, Megan is the best student she's ever had for RNA-seq analysis. And so maybe she'll give us some pointers today. I'm looking forward to hearing updates on her work.
Yeah, thank you. All right. All right. Thanks, everyone. Yeah, I'm really excited to be here today to talk about my thesis work, working to understand dormancy, maturation, and germination of Cryptococcus spores. Can everyone hear me okay without the microphone? Okay, cool. So I'm in Christina Hull's lab, and in the Hull lab, we study the human fungal pathogen Cryptococcus neoformans. Cryptococcus is an opportunistic environmental pathogen that causes severe meningoencephalitis in immunocompromised individuals. Cryptococcus grows as a budding yeast in the environment, and this is an SEM taken by a postdoc in our lab of a budding yeast. And I would like to make a special note to mention that Cryptococcus as a budding yeast is sufficiently distinct from Saccharomyces cerevisiae. We do still refer to it as a budding yeast, but it is over 500 million years diverged. It's very distinct. But so it grows in the environment as a budding yeast and will produce aerial spores under nutrient limiting conditions. And in our lab, we have been able to show that spores and yeast are able to cause disease in animal models. So we have been working to define Cryptococcus as a model spore-forming pathogen to better move forward our understanding of spore-mediated diseases. So Cryptococcus, as I mentioned, produces these spores under nutrient-limiting conditions. And as I said, we have been able to show that Cryptococcus spores are the infectious propagules in spore-mediated disease. And so I'm showing you the currently accepted model of Cryptococcus spore-mediated disease in humans, where spores are inhaled from the environment into the lungs, where they will then germinate into yeast and escape from the lungs into the bloodstream, where they can then disseminate to the rest of the body, ultimately crossing the blood-brain barrier and causing an overwhelming fungal burden in the brain. And so this is what is uniformly fatal if left untreated. So our overarching lab hypothesis is that if we're able to prevent spore-specific processes such as germination in immunocompromised individuals, we can prevent spore-mediated disease. So the work that I'm going to talk about today is focused on the basic biology of spores and how we can better understand that basic biology and develop more targeted antifungal drug therapeutics. So Cryptococcus, as I mentioned, will produce spores via sexual development. And this initiates when two yeasts of opposite mating types detect one another in nutrient limiting conditions. And those two yeasts will fuse and initiate dikaryotic filamentation. where where in response to unknown signals, they will stop filamenting and form these microscopic mushroom heads called basidium or fruiting bodies. And in that basidium, those two nuclei will fuse, undergo a single round of meiosis to produce 4 haploid nuclei here. And then those nuclei will undergo repeated rounds of mitosis and be packaged into spores that will protrude off the head of the basidium into 4 chains. Those spores will then disperse out into the environment where ideally they will find a new and more suitable environment to germinate and transition back into yeast. So germination is essential not only for spores to cause disease in humans, but it's also essential for survival of the species, because if a spore can't germinate, it can't re-enter vegetative growth. And so the work that I'm going to talk about today will expand on this model in 2 separate areas. I'm going to talk about some work that I've done to understand a maturation process in Cryptococcus spores, and work that I've done to better understand the requirements of spore germination. So spore maturation in other fungi is known to contribute to changes in germination potential, but until now, we haven't had any evidence of a spore maturation process in basidiomycetes like Cryptococcus. So for example, we know that in Aspergillus spores, maturation is accompanied by a clear physical change, an increase in melanin, and a decrease in germination potential. So we wondered, do we see something similar in Cryptococcus spores? And so in order to address this question, we've had to develop a couple of different tools to better understand the biology of spores. And one of the tools that we've developed in our lab is our quantitative germination assay. And I'm going to take a couple of minutes to describe how this assay works, because I'll be showing a couple of different plots generated using this assay. So this is a high resolution population-based microscopy assay where we take populations of spores and load them into 384 well plates, 384 well plates, and then we take images over time as spores germinate. And this works on a very fundamental principle that spores are small and oval and yeast are large and round. So we take those metrics and measure those metrics for each individual cell and plot them against each other. So cell size we determine with area, and aspect ratio represents spore shape, which is width over length. And you can see really clearly that spores cluster on the far left side of these 2-dimensional histograms, and yeast cluster on the far right. So we can see really clearly what is the makeup of a population at a given time point. So as I said, we image over a time of about 16 hours. So here's a fully dormant population where all the cells are clustered over on the far left. But after we supply a germination trigger like glucose and wait a couple of hours, we start to see the spores get more round and move up the plot, and then get larger and move over to the side. So this is an intermediate stage. And so by 16 hours, we have fully germinated cells. And we can use these predefined cutoffs that we've developed with this assay to measure specific kinetics of this process. So one of the kinetics that we're especially interested in is essentially a percent germination. So we think of this as the percent of the population, of the starting population of spores that has transitioned out of the spore state at a given time point. So time is along the x-axis, and then percent of initial starting population transitioned is on the y-axis. So I'll be showing you a couple of those different plots today. And this assay has been especially valuable for us to understand spore response to various inhibitors, spore response and germination in the context of different carbon sources, and how different mutants respond in germination. And then so I used this assay to start addressing this question of spore maturation. I isolated spores from crosses after 2 days, or what we would consider very immature spores, 3-day crosses, what we would consider slightly more mature, and then 7-day, what we've been considering a fully mature spore population. And I ran a germination assay using dextrose as my carbon source, and saw that 3-day spores and 7-day spores track really similarly. You can see that they have very similar germination kinetics, but 2-day spores are extremely deficient in initiating germination, and they experience a sufficient lag. So this told us that not only as we don't see any physical differences between these spores, but they do appear to be having some distinct differences in their germination potential. So this informed our working hypothesis that spores are undergoing a maturation process that is promoting germination competency. And what was even more exciting to us is as we teased apart this phenotype by using different carbon sources and various inhibitors, we saw that this appears to be more of a continuum, and that there isn't a single event that happens after 3 days that creates a population of mature spores, that really there's a maturation process over 7 days. And so again, I'm showing you the same plot that I had already shown you, but this is our reference germination profile that we like to refer back to of germination in dextrose. But if we switch this up and use a complex carbon source, we can see that 7-day spores fare fairly well as compared to their 2- and 3-day spore counterparts, where 2- and 3-day spores really struggle to germinate in any complex sugar. And we see similarly with the complex sugar trehalose, where 7-day spores, they manage fairly well. 3-day spores are teased down a little bit, and 2-day spores are really bad at germinating in not optimal conditions. And so this told us again that this spore maturation process appears to be a continuum, and it's conferring an advantage to spores to fully mature. And so this informed a working hypothesis that these changes in germination capacity will be accompanied by changes in gene expression that will give us a clue of what is actually going on in spore maturation. So we carried out an RNA-seq experiment where we isolated spores from 3-day crosses and 7-day crosses, extracted RNA, sent it off for sequencing, and saw that while it's subtle, there are about 150 transcripts that are differentially expressed between these 2 different time points. And so we saw that 65 transcripts are increasing in levels from 3 to 7 days of maturation. And of those, of note, 18% have absolutely no predicted function, in that they have no protein domains that are recognizable, and they have no orthologous partners outside of the Cryptococcus family. And of those, it's a small gene set, so we've been hesitant to do any Go enrichment, but we've looked at for general trends and seen that about 23% of the transcripts that are increasing in levels are involved in cell cycle processes. And of the transcripts that are decreasing, about 30% of those, again, are part of that no predicted function classification, and 19% are involved in processes involving translation. And so this informed our working model, where maturation is contributing to the establishment of germination competency. And immature spores have this transcriptome that's reflective of recent biogenesis that is not capable of quick germination as a mature spore is. So immature spores have a lower germination potential, and mature spores appear to have a transcriptome that are reflective of the preparation for vegetative growth, in that they have cell cycle components starting to be upregulated as if they're preparing for germination. And they are poised to germinate in diverse environments. And so now I've told you about the work that we've done focused on spore maturation. I'm going to switch gears and talk about how maturation contributes to the establishment of a germination competent spore and what it means for a spore to initiate germination. And so the driving question moving forward was, how does the mature spore transcriptome transition to the germinating spore transcriptome? And so we took another sequencing approach, a transcriptomics approach, where I isolated RNA over the time course of germination, so from 0 to 10 hours, collecting samples every 2 hours, with an additional 5 samples in the first 2 hours, where I collected samples every 20 minutes. All of this to get the first 2 hours in greater resolution. And so we anticipated to see the transcript levels change relatively slowly over this entire time course. We imagined we would see distinct waves of gene expression, and that we would be able to pick apart very specific processes that are extremely important at very specific stages. And we were surprised to see that we didn't see this at all. What we found was that the most dynamic changes that are happening in the time course of germination actually happen within the first 20 minutes of germination. So right now I'm showing you a principal component analysis plot of my gene expression data. And just to orient you, dormant spores are all the way over here on the far left corner, and fully germinated spores or yeast are up here on the top of the plot. And the most drastic changes happen between dormant spores and spores 20 minutes into germination. And so if we look at a heat map of these data, we can see pretty clearly why this is. maybe not on this projector, but in this plot, each row is a gene and each column is an individual replicate ordered by time. And you can see relatively clearly that between time 0 and 20 minutes, there's a clear shift in the transcriptome, and the rest of the changes are relatively modest for the rest of the time series. So this told us that there's a transcriptional program that's established within immediately within the first 20 minutes of germination. And so we wanted to understand more about what was going on in those first 20 minutes. So we carried out a differential expression analysis of those first 2 time points between 0 and 20 minutes, and broadly saw very general trends that the first 20 minutes show a very clear shift in catabolism to anabolism. And not to belabor this point any more than that, it was the most striking thing for us was that everything, nearly 30% of the transcriptome experiences an extremely dynamic change in those first 20 minutes. So then we wanted to know, how can we determine what are the most essential processes and regulators of germination initiation? Since we saw such dynamic changes, it was difficult to narrow down to what was most important. So we've turned to a set of mutants that we've been working with in the lab that have extremely interesting profiles during germination, and 2 of which I'm going to talk about today, HDR2 and ISP2. HDR2 is a transcription factor, a high mobility group regulator 2. It's part of a transcription factor family that is known to regulate developmental processes in fungi. And ISP2, which is one of those really interesting no known relatives genes that I talked about earlier, it has no orthologs outside of the Cryptococcus species complex. And so both of these knockout strains that we have in the lab actually germinate at a faster rate than wild type. So wild type is in black here, and HDR2 is in purple, and ISP2 is in green. And you can see really clearly that there's a very striking difference between the germination profiles of these 2 mutants and wild type spores. And so we hypothesized that the knockouts that we are testing with similar phenotypes would have similar transcriptomes during germination. So we took another transcriptomics approach. I isolated spores from ISP2 crosses, HDR2 crosses, and wild type, collected spores germinating at 0, 20 minutes, and 2 hours. And we were expecting to see a very similar transcriptomes. But we were surprised to see that HDR2 and ISP2 actually have very distinct germination phenotypes or germination transcriptomes. When we looked at the genes that are differentially expressed between them, only about 6% of the genes differentially expressed in the entire experiment were shared between HDR2 and ISP2. And this told us 2 really interesting things, that HDR2 function in parallel repressive pathways, and in their absence, germination can initiate faster. And then also that those overlapping differentially expressed genes are really promising candidates for future studies of germination, knockout candidates, I mean. And so all of this together is informing our repressed spore state model, where HDR2 and ISP2 are functioning in separate repressive pathways, and a germination competent spore has this transcriptome that is required to be a dormant spore and also immediately establish the germination program after a germination trigger, such as a utilizable carbon source. And that those 2 dormancy factors, as I said, are repressing those essential processes for germination until those appropriate conditions are met. So with that, I wanted to talk a little bit about rigor and reproducibility in these 2 contexts and the types of experiments that I've talked through. So you can see we've done a couple of different transcriptomics experiments, and one of the care that we've taken to make sure that this is rigorous and reproducible is that we're consistently recording all versions of reference genomes and packages used. All sequencing data is available for everyone in the lab to use, and also will be published and submitted to NCBI or another consortium once we publish. With our microscopy-based assays, we make sure to quantify thousands of cells across multiple replicate wells to make sure we're really getting a representative population. And then we make sure to hold on to those raw and processed images on external hard drives over time so that we can come back to them if we ever need to or share them with collaborators. And with that, I would like to thank the whole lab and my thesis committee, especially Anna and Ming Wei, who have done work in the lab with ISP2 and HDR2. With that, I'm happy to take any questions. Yeah.
I mean, it looks like all the transcriptome action is in the first 20 minutes, right?
Mm-hmm.
But the phenotypic shift took a lot longer, didn't it?
Yeah.
So what's going on in between there?
Yeah, that's a really interesting question. So initially we thought that we would see really distinct waves along with each of those morphological kind of intermediates. We'd see really clear waves of gene expression, but we were extremely surprised that that was not at all what we saw. We were even more surprised that it happened within 20 minutes. I guess we had some inkling that it would be rapid, but we didn't think it would be that rapid. Yeah, so what we're thinking is happening is that, and I didn't put this in here because we don't have a ton of supporting data just yet, but we're just starting to collect that, is that transcripts are stored in the cell, in the spore during maturation and they're processed, and the poly A tails are actually removed. And then upon germination initiation, poly A tails are added back on, and that's why we can detect those transcripts after 20 minutes. Because it's a surprising amount of transcription, but we're actually, yeah, it's an extremely surprising amount of transcription to happen that quickly. So we think that a lot of the lag, I guess, in what we're seeing of morphological change is probably more modulation of transcripts and like cellular contents, and that it just takes a little while before you can actually start to see any morphological change. That's what we're thinking right now. Yeah, Nathaniel.
What do you think would happen if you played with the amount of glucose that you're using or any sugar that you're using as a trigger? Because I'm imagining this process as being like, you don't want to germinate too quickly in case there isn't actually enough resources around to grow. So is there a threshold or something?
Yeah, we have, we've done those experiments. We have, the solutions that we use is it's about 2% glucose total. We've titrated down to, I believe it's like 0.0054% glucose. And what we'll see is that spores will germinate to like a specific point on the histogram, if you can envision it, and then they'll pause. So they'll use up all of the available glucose and then they'll stall. But the single dilution up from that, they can make it all the way through germination. So there is a point where they use up all the glucose in the environment and then they run out, and they just, I guess, like they just have to stop.
And do your mutants affect that pattern?
I have not done glucose limiting experiments with HDR2 or ISP2. That would be really interesting. We have also done some like spontaneous germination experiments where we do not include any carbon source, and we just let them sit in PBS for like 5 days at 30 degrees. And we actually do see germination there. So it's as if the temperature, there's many things that need to come together in order for germination to initiate. Andrew?
I was going to ask a question similar where like you give them glucose for that 20 minutes and then you take away the glucose. Does the transcription sort of reverse? Is this sort of a reversible process, or is it just like a pause? I'm curious if anyone's looked at that.
I have done some like spike and remove experiments. They're a little bit messy just by like the nature of the experiment. And since I'm moving around my like plate a lot, I'm moving around the cells. So they have been difficult to interpret. But a similar experiment that we have attempted, because we haven't done any transcriptomics to check that, but that would be a really interesting like try. Like if you initiate germination, can you remove it? And like, do they just go back to being fully dormant and hang out the whole time? But yeah, we've tried with some of our more complex sugars. They have really distinct profiles, a little bit more distinct than like dextrose. So we've tried spiking with dextrose, removing it, and then adding something like raffinose, which has a characteristic delay. And those experiments have also been pretty like difficult to interpret. But that is a really good question, and something that we have also tried to address. Linda, did you have a question?
Yeah, I was curious also with the rapid change in transcription. Have you looked at, or has anyone else to your knowledge done something similar with Aspergillus or like Saccharomyces
Yeah.
Different spore types.
Yeah, there's a couple of publications out there that look at time courses of time course transcriptomics of germination. I know there is one for Saccharomyces, there's one for Aspergillus, a couple of different members of the Aspergillus family, and a couple of other like plant pathogens, I believe. And typically they do see that really dynamic 0 to whatever their first time point is, but a lot of times those publications use like 1 or 2 hours as their first time point or their second time point. So to my knowledge, I have not seen one that has this short of a time frame. Yeah.
Is diabetes a risk factor for Cryptococcus?
I do not know. I don't believe so. As far as we know, disease progression of Cryptococcus, I would say those that are most at risk are people undergoing chemotherapy, people who have had solid organ transplants. I have not read anything about associated with diabetes. Yeah. Francisco?
Yeah, nice talk.
Thank you.
It sounds like you were using PA as selected RNA for your RNA-seq.
Mm-hmm.
So even though with early development, there's also that problem with use PA selected that you get an artificial spike because of the polymorphism. It's not reflected, right? So people are moving to use co-RNA and also so that you don't have that artifact, and also introns. I know there's not a lot of introns in this, which is good enough, but at least in the superfish, for example, introns can be used to look at new expression and those kinds of transitions, because the mature RNA is forward, and the new expression is wounded introns.
Yeah, that's definitely something that I can go back to the raw data and look at. Cryptococcus actually has a surprising amount of introns. It's like one of the most intron-rich genomes with at least fungal genomes. And to the first part of your question, we are working on putting together, like prepping everything to do a ribosome depletion experiment and supplement our poly-A selection experiment with those data to get a better sense if that is happening in our system as well. Because I think until now, we didn't really have any reason to believe that it would mimic what is seen in mammals. But yeah, I think this is reason enough to think that it could be happening. Yeah. Yeah, I'll have to look into how to do that. Oh yeah, okay. Yeah, that'd be really great. Thank you. Okay. Great, thank you.
Thank Megan again.